So good afternoon and welcome to this session on biological control. And we have entitled that the importance of augmentative biocontrol and plant microbiome function for plant health. Actually, we were two groups coming up with almost the same title, and we decided to coordinate these and have two parts, part 1 and part 2. So this is part 1, and tomorrow we will have part 2, and that will be chaired by Monica Höfte and Claire Pruchon-Combarat. So that will be tomorrow, 10:30. So very briefly, why do we have this title, the importance of augmentative biocontrol and plant microbiome function? And that's because if you look at augmentative biocontrol, meaning that you use a single organism or a few organisms together as biocontrol agents in consortia and to control diseases, that took off in the 1970s with intensive research and also development, and it has been going on since that time, and now there's a lot of commercialized products on the market, and it has really been starting to be used commercially and in agriculture. And then if we go to about 2 decades back, we had what we at that time called the next generation sequencing, and that could be used for describing the microbiome in different parts of the plant, inside, outside, in wounds and on leaves and so on, to find out what do we really have of microorganisms related to the plant and to the root system and also in soils. And to begin with all these techniques, sequencing techniques, it started out being very descriptive, so we could really find out what is there, but what are they doing, these organisms? But I mean, that start of microbiome studies really gave new information about a lot of different organisms. And this is the other side of biocontrol now, that you can look into what is the function, and that's what we see now, that we are trying to put function into biomes and how does that relate to plant health. So then we think we have these two lines of research in biocontrol that need to maybe also to talk more together to find out what have we learned from the augmentative biocontrol that has been going on for many years and what is coming out of the new biomes, and that we hope that there will be a discussion about aspects of that. And for that we have these two sessions, part 1 and part 2, and we are 3 chairpersons, so I would like to introduce my 2 co-chairpersons, Sabrina SARROCCO from University of Pisa in Italy, and David Collins from Copenhagen University in Denmark. So we have the next speaker, and that is Dr. Mukesh DUBEY from Swedish Agricultural University of Swedish University of Agricultural Sciences. And Mukesh has been working almost 14 years with biological control, I'm sure, and mainly with interactions in biocontrol. And recently, a few years ago, he started to see what role do small RNAs have in mycoparasitic interactions. And I think that will form the basis for the new future, new mechanism. What role do these small RNAs have in mechanistic interactions in biocontrol? So, what is yours?
Thank you, Dan. Thank you for the introduction. I have to change it to myself. Or
sorry, I was going to do that. You just press. You got yours? Yeah.
No. Okay. So good afternoon, everyone, and thank you for the committee for the invitation, and so I could share some of our ongoing work here. And I will talk about small RNA-mediated mycoparasitic interactions. I will start very briefly with a brief introduction, probably, that small RNA are non-coding RNA molecules, and they mediate gene expression regulation by a process called gene silencing and RNAi. And if you see this process that here, basically there are two main components in this, dicer protein and ergonard protein. Dicer protein, they act upstream in the process, and then they cleave the double-stranded RNA or single-stranded RNA with hair loop structures to produce siRNA or miRNA, respectively. And this small RNA, they are unwounded, and then they are loaded on a protein complex that is called RISC, and where ergonard protein, which is a main part of this RISC complex, that guided by this small RNA to the gene target, where they degrade the gene target into small pieces. And this small RNA-mediated degradation product can be cloned by a method that is called degradome. I will come back to this in a few minutes. So, and the recent discovery, at least in the last decades, it has been a lot of work going on mainly focusing on the role of small RNA in host microbe interactions. And there is quite a lot of evidence coming up that small RNA can mediate interaction between host and pathogens, leading to pathogenesis or mutualistic interaction. However, that is very well summarized in this image, but the role of small RNA in mediating mycoparasitic interaction is not very well studied, or it's very limited study available on it. So as we all know, probably this is a biocontrol session that the mycoparasitism it's a complex process where you have, as a mycoparasites, they need to regulate their gene expression pattern to produce a range of chemicals like secondary metabolites and enzymes to parasitize and kill the mycohost. So what we did, we hypothesized that during this process, small RNA can mediate, this gene expression regulation. So that was our main hypothesis in this. And then to test this hypothesis, we used a fungus that is a clone of Trichoderma. It's well known for its biocontrollability. And the main thing in this, that it can also parasitize, it has a broad host range, and it can also parasitize plant pathogenic fungi, for example, Fusarium graminearum and Botrytis cinerea, even under in vitro conditions, even in plate. So what we see in this image here, the Clonostricus rosea in white, and then it can easily overgrow and kill the Fusarium graminearum here in this middle image, and then the right side you can see Botrytis cinerea parasitized by Clonostricus rosea. So what we did, we used this fungus as a model to study this phenomena, the small RNA phenomena. And first, we have a genome sequence. So we searched the genome sequence of Clonostricus rosea and found that it has RNAi machinery gene available. For example, it has 2 genes coding for dicer proteins and 2 genes coding for ergonard protein, but also it has 3 genes for RDRP, RNA-dependent RNA polymerase. So what we did, we used these 2 fungus, plant pathogenic fungus, Botrytis cinerea and Fusarium graminearum as 2 mycohosts. And then we used Clonostricus rosea, and we set up a dual culture interaction assay. And then we harvested the mycelium from the interaction zone, and then we sequenced the small RNA and degradome, and also the transcriptome during this interaction. And then we also went one step further. We created knockout mutant of dicer 1 and dicer 2 in Clonostricus rosea, and then analyzed the phenotype and also the small RNA profile and also the transcriptome profile and then compare with wild type and mutant. And we also used this dicer mutant, to identify the microRNA, we also use this data to see if production of small RNA or microRNA is Clonostricus rosea is dicer dependent or not, because in Neurospora, dicer independent mechanism of small RNA production is also known. So what we did, so the first part is like analyzing small RNA and degradome profile. So from this experiment, we got 3 take-home messages that I can try to summarize you. The first take-home message was that a higher number of small RNA, they were downregulated during the mycoparasitic interactions compared to self-interaction control. That's what you can see in this bar diagram. So meaning that, that a small RNA, they are negative regulator of the gene expression. So meaning that if the small RNA are produced in higher amount, their gene target would be degraded. So there is a lower amount gene suppression. So there would be gene there, and we thought that a lot of mycoparasitic related genes probably is that were suppressed in the self-interaction control. They were upregulated when there was a mycoparasitic interaction. The second take-home message from this work was that if you see this bar diagram carefully, if you see right side, this is in blue, you see Clonostrachus rosea interacting with botrytis, and then in the right side, it's data from the Clonostrachus and Fusarium interaction. So it's not very high, but still the response towards Fusarium was relatively more compared to response towards botrytis cinerea. So we thought that maybe Clonostrachus has some mycohost-specific response, and which was confirmed from this data, where we used WGCNA to run a correlation analysis, and then with this analysis, whole small RNA that were differentially regulated, they were separated into 13 different modules. So if you see, for example, module ME1 on the top, so you have, and then here it's red to blue, it's like a Spearman correlation analysis. Minus means that is in the blue, it's a negative correlation, and the plus one, it's in the red, it's a positive correlation. So it seems, for example, in ME4, you see there is a self-interaction control, you have a minus, and then here interaction with Fusarium, you have a red. It's like, and then in between, it's no significant difference when Clonostrachus was interacting with botrytis. So I will not take more time to explain this, but you have to trust me that there was a Clonostrachus rosea responded to botrytis cinerea and Fusarium graminearum. This response was mycohost dependent. So it seems that Clonostrachus could recognize the host and responded accordingly. Now as you said, as I told you before, that we use digidome sequencing to identify the gene target. And in this case, we could identify 282 gene target from 201. Sorry, we identified 201 the differentially expressed gene target for 282 small RNA in Clonostrachus rosea. And when we did gene enrichment analysis, we found that majority of this gene target were related with membrane proteins or carbohydrate and lipid metabolites, and also small secreted proteins. So probably in this class of proteins, somehow they are also related with the mycoparasitic ability in different mycoparasites, including Clonostrachus rosea. So now we know that there was a strong indication that Clonostrachus rosea uses a small RNA to regulate gene expression. And then we deleted the DICER1 and DICER2 mutant in Clonostrachus. And then we found that the DICER1 was redundant for its mycoparasitic or biocontrol ability. However, DICER2 mutant has a reduced ability to mycoparasitize botrytis cinerea under in vitro condition. So that's what you can see in this image, that wild type Clonostrachus can overgrow botrytis, but DICER2 mutant, this is the growing front. It has a reduced ability to overgrow, and this was significant here. And we found similar result for its biocontrol ability against Fusarium graminearum. So if you have a control, you have a lot of disease, but if you treat seed with Clonostrachus wild type, there's a significant reduction in the disease development. But then if you have a DICER2 mutant here, then it has the level of disease went up, meaning that DICER2 mutant had reduced ability to control Fusarium graminearum. So in order to, so we also found another phenotype that was even more interesting, that DICER2 mutant had a reduced ability. If you see here these plates, Clonostrachus gives you very nice color if you grow this fungus on PDA. But if you see DICER2 mutant, the production of yellow color was reduced. So we did a whole metabolome analysis of Clonostrachus wild type, to DICER mutant, and also the complementation strain. And then there is a different set of metabolites that were affected in the DICER2 mutant. Just to show you one example, if you see this wild type in the left side, and then DICER mutant in the middle, and this is, basically this different metabolites related with the sorbicillin. Sorbicillin is a polyketide as known for its for yellow pigmentation, and it's also known for its it's also known that it has a fungistatic ability. So it seems that dicer2 had a reduced ability to produce the sarbicillin, and then in the complementation strain, the function was restored. So, and we know we had the genome sequence, so we figured out the sarbicillin biosynthetic gene clusters, and we found that the core gene, that is polyketide synthase 2 and PKS22, and some other accessory genes, the expression of those genes was significantly downregulated in this biosynthetic gene cluster. So it's kind of meaning that, however, I can say one thing, that we did not find the microRNA with a direct binding site to this gene. So that is still under a matter of investigation. In order further to explain that if deletion of dicer2 has an effect on small RNA profile or gene expression analysis, then we compare the small RNA of clonostricus wild type and deletion mutants. So deletion of dicer1 has no effect on small RNA production or size classes, maybe a little bit, but you see here a significant reduction in small RNA size class of 80 to 22 nucleotide base pair. So indicating that dicer2 is playing a major role in small RNA production. And this is also very well reflected here in the...
Can you come to a conclusion?
Yes, it's also very well reflected in its gene expression profile. So maybe I can a little bit speed up here. So one important finding from this work was that we managed to identify 62 microRNA in this work, and then out of this 61 microRNA, we found 12 microRNA that were dicer2 dependent. And then again, as I told you in the beginning, that we had harvested the samples from interaction zone, so we could also sequence the transcriptome from Botrytis and Fusarium, and then we used this data to identify the cross-species gene target of clonostricus microRNA in Botrytis and Fusarium. And using this data, we could identify some genes that has a cross-species gene target in Botrytis and Fusarium. But at the moment, we are working on it to prove if this is true or not. If the similar result we found against Fusarium. So in summary, this is and also a perspective that what we think that if you have RNAi functional, then it can regulate the gene expression pattern, which in turn affect the mycoparasitism and biocontrol, and help the fungus to do this function. But if you have impaired RNAi, which is at the bottom of this image, then it affects its mycoparasitic and biocontrol ability. Of course, there is a lot to do in this field, so we are working on it to functionally characterize the small RNAs. And with the last slide, then I would like to thank our collaborators and the funding agencies.
Thank you, Mukesh.
There's been one question I've answered it. There's one question if we don't have time. We have one question which is asking about the mode of action of clonostricus against its target fungi. And for example, antibiosis coiling around the hyphae. And I'll so either you or Dan can answer the question.
I think it's a very broad question.
Yes.
And I think here we focus on the role of small RNAs as a new paradigm in biocontrol interactions, and I think that's the point you wanted to make.
Yeah.
That they really have a big role in not just antibiosis and enzymes and so on competition, but they are involved in all these processes in fungal interactions in biocontrol.
Yes.
Yeah.
Yes.
I think your time.
Thank you very much.
Has run up. Thank you very much.
Okay, so it's a pleasure for me to leave the floor to the next speaker, Professor Enrique Monte from the University of Salamanca, Spain. I suppose all of you already know the huge amount of work made by Enrique on Trichoderma as a biocontrol agents against plant pathogens, as inducer of resistance, to improve the tolerance to abiotic stress, and now let's hear something about non-conventional use of Trichoderma fungi. Please, Enrique.
The presentation is here. Okay. Yes, okay. Good afternoon, everybody. Thank you very much for your words. And thank you very much for your invitation, Sabrina, Dan, and David. Then I'm coming from Salamanca. Salamanca is in northwest Spain, near the Portuguese border. And I'm coming from a very old university since 1218. It's more than 800 years old. Then, who is Trichoderma? Trichoderma is a filamentous fungus, anamorph, mitotic reproduction, with a perfect state, sexual reproduction in the Ascomyces hypocrea. We have recently published an article in Nature Review Microbiology describing the role of Trichoderma in agriculture as both a biocontrol agent and a biostimulant. I'm going to talk about the biocontrol activity against non-conventional targets such as insects and nematodes. But initially, the mechanisms of action of Trichoderma as biocontrol agents are the use of cell wall degrading enzymes against fungi and or myces, and also the use of secondary metabolites, most of them with antibiotic activity against fungi or myces and bacteria. This picture belongs to my PhD thesis in 1986. I isolated the Trichoderma strains against the pathogens of sugar beet and pinov, mainly Fomavite and Rhizoctonia solani. However, these mechanisms were described in the first half of the last century, the antibiosis and mycoparasitism. Then, today I'm going to talk about this characteristic of Trichoderma as biocontrol agents of nematodes and insects. Nematodes and insects have a chitin in their body. The insect cuticle and the peritrophic matrix are made by chitin, and also the eggshell of the nematodes. Trichoderma is able to secrete glucanases, chitinases, and proteases. And then these targets, or these animals, nematodes and insects, can be targets of these enzymes. Then, long time ago, in 2004, we purified and cloned the gene, the PRA1 gene, with nematicidal activity. We started checking the antifungal activity against Colletotricum acutatum. It was very, very easy to control Colletotricum with this protease. And with the purified protease, we applied this extract to eggs of Meloidogene javanica, and we destroyed the eggs and could control the disease produced by this root node nematode. Other groups have checked the chitinase activity of different species of Trichoderma. In this case, Trichoderma viride is able to destroy the peritrophic matrix of the silkworm Bombyx mori. In a One Health approach, Trichoderma is a protagonist because the chitinases produced by Trichoderma can control the mosquito Aedes aegypti that is a vector of human diseases such as dengue, Zika, or yellow fever. Also, volatiles released by Trichoderma, cis-pentylpyron and 1-octane-3-ol, can be purified, extracted from the volatilome of Trichoderma, and they can be applied pharmacologically to the diet of insects, Spodoptera frugiperda, the corn and cotton bollworm. And these larvae, then they are not eating in the normal way. These volatiles have a repellent action, a repellent function, and as a result, the insect has a reduced development and an increased mortality. Trichoderma also release volatiles that can be attractors of pest enemies such as predators and parasitoids. In this study, developed by Contreras Cornejo in Mexico, the volatiles of Trichoderma, in this case 6-PP, 6-pentylpyron, can attract the wasp, Compoletis sonorensis, that can eat the eggs of Spodoptera. Then, Trichoderma, in interaction with the plants, can be used as biocontrol agent, indirect biocontrol agent, because Trichoderma can stimulate the defense of the plant against fungi or myces, bacteria, viruses, insects, nematodes, and even insects. But Trichoderma can also, in the absence of pathogens, promote the seed germination, promote the growth of the plant, and enhance the tolerance of the plant to abiotic stresses. This feature is marketed as biostimulant. Then, as a result, when Trichoderma controls the pathogens, the plant keeps in the transcriptomic memory the stimulation of the immune system, and this phenomenon is known as priming. Then Trichoderma can prime the plants to defend itself against a subsequent stimulus. But this stimulus can be biotic or abiotic. And finally, after this activation of defenses of the plant are kept in the transcriptional memory, this effect, the Trichoderma effect, can be transmitted to offspring, to the next generation. Then these responses of the plant to Trichoderma can be inherited. Then the priming induced by Trichoderma against abiotic and abiotic stresses has an undulating profile. In the first hours, it's very intensive, but disappear after 3 weeks, more or less 3 weeks. And there is a continuous change or balance of the salicylic defense responses or the jasmonic and ethylene dependent defenses. We have checked this behavior in 2 different crops, so different, such as tomato or wheat. The beneficial effects of Trichoderma are most apparent in stressed plants, but not also against the pressure of a pathogen, but also, for instance, water stress. Here is the demonstration of Trichoderma-induced priming against the nematode Meloidogyne javanica, and we developed, since this is a pathogen of the roots, we developed a split root system. We put in the control water in one side, then we separate the roots of the tomato plant into pots. In the control, we applied water, in the treatment water Meloidogyne, and in the treatment with Trichoderma in one pot and Meloidogyne in the other pot. And we put also a control of Trichoderma. In absence of the nematode, we could see growth promotion, but in the presence of the nematode, we identify the systemic defense in terms of reduced number of galls and adult nematodes. Then, what happened during the life of the plant? Then Trichoderma-induced priming is adapted to plant needs. In the first hours, during the invasion of the nematode, the plant activates the salicylic acid-dependent defense. 2 and 3 days later, the jasmonic acid and ethylene-dependent defenses are activated, because this is the period in which the galls are developed in the roots. And finally, after 1-2 weeks, salicylic acid is activated again, because this is the moment of the egg laying and the liberation of the juveniles of the nematode. Then, in this experiment, we are not nematologists, and we had to keep the nematodes in the pots, and then for further experiments. And the plants that survived in the previous experiment gave fruits, and we collect the seeds. We sow the seeds from every treatment, and those from the treatment with Trichoderma and water, when they grew 15 days later, we could see a 30% of increased growth. But those plants that survived to the attack of Meloidogene from the treatment Trichoderma troviride in one pot and Meloidogene javanica in the other pot were tiny. Then the phenotype was very ridiculous, but the defenses were highly activated. These plants had both salicylic acid and jasmonic acid dependent defenses. Both were activated. When we infected these plants, these tiny tomato plants, with the nematode was unable to produce disease to these plants because they have very, very low growth but a high defense, very activated immunity. Then finally, we have seen a last novel mechanism against insects. This is a new pest biocontrol mechanism induced by Trichoderma. Trichoderma treatment modifies the plant metabolome. We applied Trichoderma to the root, and the metabolome of the leaves is modified by this treatment. But the larvae, in this case Septoria, a good microbiome of leaf-eating larvae was modified as well. Then, when the good microbiome of the larvae were studied, we could see that the dominant bacterium, Enterococcus caseiliflavus, was modified because reduced its level in the good microbiome of Spodoptera litoralis. Plants treated with Trichoderma had lower levels of this Enterococcus, and as a result, we could see a reduced insect survival, a reduced development, and lower longevity. As a summary, Trichoderma is the world's most widely used biological control agent for fungi and oomyces. As an antagonist, Trichoderma can simultaneously deploy more than one biocontrol mechanism. Trichoderma possesses the largest arsenal of fungal cell wall degrading enzymes, chitinases, glucanases, a high amount of proteases as well in the kingdom fungi. The major secondary metabolites of Trichoderma with antibiotic potential are pyrons, terpenes, and peptidyls. But they are not the only ones, because Trichoderma produce more than 120 different molecular types. Trichoderma has nematicidal potential via chitinases and proteases. Trichoderma has insecticidal potentials through chitinases, proteases, and volatiles with repellent function. Trichoderma also produce volatiles that attract parasitoids and predators of insect pests. Trichoderma exerts indirect biocontrol priming in the transcriptional memory via the plant that is adapted to plant's needs. Trichoderma priming is inherited by offspring, and Trichoderma modifies the metabolome of the plant and influences the microbiome of the insects that feed on it. And finally, thank you very much. This is my group of the University of Salamanca Institute for Agribiotechnology Research, and the only person that is not in the photo, Isabel Vicente, is because did the PhD in Pisa with Sabrina, and now is in Napoli with my colleagues from the Federico II. Thank you very much.
Thank you, Enrique. We have several questions from the audience, but I think we have time only for one or two. So one is no name, unfortunately. Are some strains of Trichoderma more specialized than others in controlling nematodes? And could Trichoderma be breeded to make it more effective?
Whose Trichoderma is more effective? Yes, then. We are involved in the project 400 Trichoderma genomes. And in this project, the bioinformaticians have built a phylogenetic tree with the genomes. And then this very, I think, very definitive phylogenetic tree. And we have seen that strains involved in biocontrol belong to two major groups in Trichoderma, the group Harsianum and the group in which are involved, the group Trichoderma, the cluster Trichoderma, Asperellum, Asperelloides, Atroviridae. Then biostimulation is spread along the genus. Then the best biostimulation strains for plant growth promotion are in any group of Trichoderma, but the most important biocontrol agents belong to these two groups. Then I would select in the Trichoderma cluster of the phylogenetic tree the most effective strains of Trichoderma. If you ask me, which is the best species of Trichoderma? To me, Trichoderma asperellum.
Thank you very much.
So I would like to introduce Magnus KARLSSON. He's from the Swedish University of Agricultural Sciences, and we have been working together many years, and he has a very interesting talk now, not looking at biomes, but going the other way, looking at intraspecific variation within a species of biocontrol.
Thank you very much, Dan. So, we can start with this picture. When we think about biocontrol, we can divide it into 4 different types. For example, we have classical and augmentative biocontrol, which involve rearing of specific agents for release, either for permanent or temporary biocontrol. And one challenge here is that you typically see a lot of high strain-to-strain variation in biocontrol efficacy. On the other hand, we have conservation and natural biocontrol, which involve working with the resident agents, with or without intervention. In this case, we have challenges with the sporadic appearance of diseases pairs with soils. So we've been thinking about this during the recent years, and one possible idea that could potentially explain both of these phenomena is local adaptation that drives population differentiation of the biocontrol organisms. In our case, we work a lot with hyperparasites. And during this lecture, we will explore this idea a little bit. We will look at questions such as, is there strain-to-strain variation in biocontrol-related traits within individuals of the same species? And if so, to what extent? And what is the underlying mechanisms resulting in this strain-to-strain variation? How can we also use it for implementation of biocontrol? So in our case, we worked a lot with this fungal biocontrol agent, Clonostacis rosea, which Mukesh DUBEY introduced previously. So I can just repeat that it's a very aggressive necrotrophic mycoparasite, attacks other fungi and kills them, and uses them as nutrient source. But it also has several other properties that makes it suitable as a biocontrol agent. It has a certain saprotrophic capacity and can live in soil. Its rhizosphere competent and can colonize the root system, in this case, colonization of tomato roots. And more recently, also shown that it, in some cases, can parasitize nematodes. So nowadays, we more look at it as an ecological generalist rather than a pure mycoparasite. In order to address this question, we started by collecting a collection of Clonostacis rosea strains. We have about 120 different strains of the same species collected from all parts of the world. And we can then screen this strain collection for different type of biocontrol-related traits to look at the distribution of these traits within the species. We would also like to try to connect these phenotypes with the genotype. So therefore, we performed whole genome sequencing using Illumina technology of 72 of these strains. So we can start to look at some of these phenotypes. We can start with biocontrol, and using this very simple SAND test, we screen this collection of strains for biological control of Fusarium foot rot disease caused by Fusarium graminearum. If we look at the result, we can see that there's indeed a large variation in biocontrol efficacy between strains. On one hand, we have strains that provides a very, very good level of control when applied as a seed treatment. But on the other hand, we also have several strains that does not give any significant difference compared with the Fusarium control, so they do not have any biocontrol property at all, even though they are members of the same species. So this was biocontrol, and we can look at in vitro antagonism, in this case production of nematocidal compounds in liquid cultures. Basically each strain was grown in liquid culture for a specified number of days, the mycelia was removed, and then the liquid was re-inoculated with root lesion nematodes, Pratelenchus penetrans, and then we assessed mortality over time. So it's a similar pattern. Some strains produce different type of compounds or enzymes that provide a very high mortality of the nematodes, up to 60–70%. But on the other side, we also have strains that actually to some extent lowers the mortality or rescues the nematodes compared with the non-inoculated media control in black in the middle. So again, there's a big variation between individuals in this trait. We can also look at mycoparasitism. In this case, it's measured as rate of mycohost overgrowth. So basically, dual plate assays between, in this case, Clonostachys and Fusarium graminearum. And then after they physically meet, then Clonostachys overgrows and kills the mycohost, and then we can measure the growth rate in overgrowth. I think this data is also normalized for the intrinsic growth rate of the individual strains. Fusarium is a very strong antagonist, so some strains are actually not able to overgrow it at all, but some strains on the other hand is very efficient in overgrowing. So we have a large variation in this trait. We can do the same thing with another pathogen, Aphanomyces certicius, an oomycete. This is not a strong antagonist, so the differences is not as strong, but you still have significant differences in overgrowth between strains. And if we make a comparison between these two data sets, then we see that we have a very low correlation and it's not significant, indicating that different strains are good at overgrowing Fusarium versus Aphanomyces and vice versa. This further indicates that there may be some level of specialization for the different individuals, and also suggests that you have different underlying mechanisms of the interaction with the mycohosts. And then we can take a closer look at what possible mechanisms that this could involve. We did an interaction transcriptomic study using these dual plate assays, and then we confronted Clonostachys with Fusarium, Orbotrytis, or a self-interaction as a control. And then we harvested the interaction zone 24 hours after contact and look at gene expression. In this case, we used very stringent statistics. So in the end, we only identified 41 genes that were differentially expressed. 60 of these were membrane transporters, but also genes involved in second metabolite biosynthesis and some carbohydrate active enzymes. If you dive into this data set a bit more detail, it looks like this. You can see that there was only 6 genes that was induced against both pathogens. They involve, for example, 4 membrane transporters transporting sugars and small organic compounds probably involved in nutrient uptake. What's more important is that it seems that you have 2 large gene sets that are induced specifically either against botrytis or against Fusarium. In the case of Fusarium, we have different type of membrane transporters, but also secondary metabolite biosynthesis genes such as cytochrome P450s, polyketide synthase, and non-ribosomal peptide synthetases. And in the case of Fusarium, then we have the same type of genes, but the identity is completely different. So we have genes involved in secondary metabolite biosynthesis and the long range of different type of membrane transporters. In this case, many of them are involved in drug resistance and drug efflux. So again, the take-home message from this is that it seems that Clonostachys has the ability to sense the identity of the mycohost and modify its transcriptomic responses accordingly. And this is also something that Mukesh DUBEY talked about. We see this also when we look at the small RNA responses in this fungus. It also implies that the interaction mechanism of Clonostachys differs depending on the mycohost identity. So far, I've only talked about the interaction between the biocontrol agent and the pathogen, but for the rest of the talk, I will swap and talk about the interaction between the biocontrol organism and the plant. In some cases, Clonostachys rosa can also promote growth in plants, in this case wheat. In this experiment, we simply mixed in Clonostachys conidia in soil without a pathogen and planted wheat seeds. And also in this trait, we see a certain level of variation between strains. In this case, it was 10 strains that gave a significant increase in shoot length of the wheat plants. We also assessed different parameters like shoot fresh weight and shoot dry weight, but the results are very similar. When it comes to the data on shoot fresh weight, we actually managed to make use of the genomic data in the sense that we managed to connect the phenotypic data with the genotype in a genome-wide association study on the biocontrol fungal side. So in this case, we identified two mutations, two SNPs in one region of the genome of Clonostachys that was segregating with this plant growth promotion trait. And these two SNPs were sitting in the middle of a transcription factor that is now under investigation in our lab. Finally, in the last part of the talk, I will shift the focus from the biocontrol agent to the plant side. And the idea behind this is that when we work with disease resistance breeding, then we make use of the fact that certain plant genotypes, plant individuals, are able to defend themselves more efficiently than other genotypes against plant pathogens and plant diseases. And we use this for disease resistance breeding. But in this case, we've hypothesized that plant genotypes may also differ in their compatibility with beneficial microorganisms and differ in their ability to benefit from these interactions. So in this study, we used biocontrol of Septoria tritici blotch in wheat caused by summer Septoria tritici using Clonostachys rosa. And this was based on many years of field trials where we saw that Clonostachys can provide a significant level of control when applied as a foliar spray. This is done by our PhD student, Siddhant Chowdhury. So he took more than 200 different winter wheat genotypes and screened them in a biotron experiment with two treatments, either only applying the pathogen, Septoria tritici, and looking at disease development, or the pathogen and Clonostachys rosa and looked at disease development. Here's one illustration of the phenotypic response of the wheat genotypes. To make a long story short, I can just say that some wheat genotypes responded more positively to the biocontrol treatment than you would expect by chance. And vice versa, you had also some genotypes that responded more negatively than you would expect. So there seems to be some preference or some differences between wheat genotypes, how they respond to this type of beneficial microorganisms. And then one reason for choosing this particular set of wheat genotypes was the fact that they were already genotyped using a 20K SNP chip. So we can immediately go ahead and try to correlate this phenotype with the genetic makeup of these plants. So we made a genome-wide association study of these traits, and if you start by looking at disease resistance, that's the data where we only apply the pathogen. And then we can see that there are two regions of the genome that is segregating with this trait, located on chromosomes 2b and 3b. But then if you do the same thing with this compatibility trait, then we get a different view. In this case, it was also two regions of the genome that segregated with this compatibility trait of the plants, but they were located on chromosomes 1d and 6b. So one conclusion seems to be that this disease resistance and biocontrol compatibility seems to be inherited at different chromosomes. So theoretically, we may think about breeding plants both with high disease resistance and high compatibility with biocontrol agents. We can also go into the genome and look at gene candidates. In this case, very close to these SNPs, we have things like a mechanosensitive channel protein, MUB-like transcription factor, and 2 different disease resistance proteins that could be candidates to work on in the future. So some conclusions. We can see that we have a typically very high phenotypic variation in biocontrol traits between different individuals of the same species, Clonostachys rosea, but we also have a high variation between wheat genotypes. Typically, we have a very low correlation between the different traits, indicating different underlying mechanisms, but also indicating that you have a certain level of specialization among the different individuals. And we start to accumulate quite a lot of information about what genes and genetic markers that is connected with this variation. So then if you look at some perspectives, on the negative side, it may be difficult to find optimal strains with a broad activity against many diseases. But on the positive side, this type of information can really help us to select optimal strains. We can also think of choosing optimal biocontrol agent, plant genotype by genotype combinations with optimal activity. We can also really start to think about setting up breeding programs of the biocontrol agents themselves, also aided by the genetic markers that we can have to choose to select progeny. We can also breed crop varieties for high compatibility with beneficial microbes, and this is something that we currently are doing together with a Swedish breeding company on winter wheat. And by that, I will thank all of you for your attention and acknowledge my co-workers and also the funding agencies. And I will be happy to answer some questions. Thank you.
Okay. Thank you, Magnus. And there's some questions for you. Oh, now it's coming a lot of. There's one here. Can you imagine to do joint GWAS in both plant and biocontrol together for disease resistance? I think maybe you can clarify it a little bit.
I'm sorry?
Maybe you can clarify that if you can imagine to do joint GWAS on both plant and biocontrol together for disease resistance.
Yes, absolutely. I mean, this experiment is done separately, but it's absolutely possible. I think for the wheat work, then it's definitely possible. We work typically with a set of more than 200 genotypes that gives enough statistical power. I think on the fungal side, then we're still limited by the fact that we only have 60-70 genomes, so the 60-70 strains that we can use. And for most biocontrol traits that are very complex, then what we can see that we may not have enough statistical power in the analysis. There's only a few analyses for some traits like the fresh shoot weight fresh shoot weights that were able to map down some genes. So I think that in order to really make use of it, we need to have more strains. But of course, it also depends on the complexity of the trait itself.
There's another one from Gabriele Berg here. What is known about epigenetics traits in Clonostachys strains? Can you say something about that?
Yeah, on a genetic makeup or genetic traits.
Epigenetic traits.
Oh, epigenetics. Yeah, we don't. Yeah, it's a short answer in a way. We don't know so much, but this is something that we're really looking into now. It's actually started with the work of Mukesh DUBEY with small RNAs, and this, of course, goes in that direction. And I mean, this data, we look at phenotypic data, and then we assume that this reflects the properties of the strains. But then, of course, as we saw during these presentations, it could also have these epigenetic changes that influence it, but also this bacterial components. We know that many of these fungi can also carry intracellular bacteria, and then, of course, these aspects would then give increase the background noise in the analysis. But this is definitely something that we are currently looking into.
Then there's one that you should just give a very brief reply, maybe. What collected strains of Clonostachys rosea from different countries around the world, or your collected strains, did you obtain legal permits for assessing biological biodiversity of local level, at local level?
Yes, so all of these strains, they are collected from culture collections, and they were all collected before 2015, when the Nagoya Protocol was ratified. So all of this we have followed all the regulations. In newer projects, then of course for example, now we have a program with Bolivia with Trichoderma, and then of course we contact the relevant authorities before doing any collections. So yes, we are aware of all these international conventions on sharing.
Magnus. Yeah, we don't have more questions.
Yeah,
Thank you very much.
Yeah, thank you.
And yeah. And at the end, there will be possibilities for a few extra questions to the other speakers, because we got some in here that we didn't post. So. We will have a next speaker, and Sabrina will introduce.
Okay, so we can move to the last speaker of this session. It's time for Mudassir IQBAL from the Swedish University of Agricultural Science in Sweden. And he will talk about the application of Aureobasidium pullulans against several pathogens on strawberry.
Thank you, Sabrina, for nice introductions. First of all, I would like to say thanks to the organizing committee of this session for providing me an opportunity to talk about my research. So today I will talk about the Aureobasidium pullulans, which is upcoming biocontrol agent against several diseases of the strawberry fruit crop. So we all like strawberry. So it's a nutritious and high-value fruit crop. When we look at the productions, the highest proportion is cultivated in Asia, 46%, which is followed by America, and in the Europe is contributing 20% of the total productions of the strawberry. When we look specifically in the Sweden, it is cultivated almost 2,007 hectares and corresponds to almost 16,000 tons yield per hectare. However, this potential is much lower because of the attack of the different diseases which is attack on this fruit crop. The most important disease is the gray mold on the strawberry, which is caused by the Botrytis cinerea. Also the fruit rot, which is caused by Trichoderma acutatum, and also the crown and root rot, which is caused by Phytophthora, which is an oomycetes. So in order to control these diseases, synthetic fungicides are being used. However, negative impacts on the environment and the biodiversity, development of the resistance against these fungicides and the banned chemical activities, and the high cost. So we need some alternative strategies and at the same time the sustainable measures to control these diseases. So one method could be the biocontrol by using the Aureobasidium pullulans, which is mainly present in the phyllosphere of the strawberry crop, and also it's present in the temperate zones. So we asked ourselves that if we isolate this Aureobasidium pullulans from the strawberry crop, can we use this as a biocontrol agent against the several diseases in the strawberry crop, just against like gray mold, anthracnose, fruit, and the crown rot? And if this is true, then which mechanism they are employed to control these diseases? So to answer these questions, we performed many experiments from the in vitro antagonism assays, greenhouse, and the field trials as well. So I will show some of them, the important findings. This is a simple test as the dual plate assays, where we tested, we grow the Botrytis cinerea and the Trichoderma acutatum on the different media, PDA, pepton, and the japacdos, which varies in their carbon sources and the pH values. And these pathogenic fungi we challenged against the different strains of the Aureobasidium pullulans and let them grow for 5 to 7 days. And then we measured the radial growth. And we see that that Aureobasidium pullulans approximately 35% reduced the growth of this pathogenic fungi compared to the control treatment. And the similar pattern was observed against the Trichoderma acutatum. So we did another experiment based on the culture filters. We produced the culture filters of different Aureobasidium pullulan strains in different nutrient media for 2 weeks almost. And then we added the agar plug of this Botrytis cinerea and the other pathogenic fungi, Trichoderma, and let them grow for 5 to 7 days. And then we measured the dry weight of the mycelia. And we have seen that Aureobasidium pullulans culture filters inhibited the mycelial biomass approximately 90% compared with the control treatment. This similar pattern was observed against other pathogens. So in this media, there will be lots of metabolites, could be the proteases and the other inhibiting agents. So we did the metabolomic analysis, high-performance liquid chromatography, mass spectrometry, and we observed that in the culture filtrate of Aureobasidium pullulans, different peaks has been observed. And when we studied these peaks, we came that they have contained lamocins and exophilins, and we also find potentially 2 new exophilins metabolites. This is previously has been known that they have antibacterial activity, but it was not known that they have an antifungal activities. Recently, we are working on it to purify it and to scale it up. So we did some in vitro antagonism assay on the detached fruit assay against the Botrytis cinerea. In this experiment, we collected the green strawberry fruits from the fields, washed them with the water, and then scrap it close to the neck and added the spore suspensions of the Botrytis cinerea and the Aureobasidium pullulans in these combinations, as we see on the screen. And then after 2 weeks, we observed the disease severity index, and we have seen that pullulans either apply alone or in combinations, or the pre-application of Aureobasidium pullulans, they reduce the disease severity when we apply the Aureobasidium pullulans. We did the greenhouse experiment against the root rot, which is caused by Trichoderma acutatum. We applied this Aureobasidium pullulans and this Phytophthora spore suspensions in the following combinations, and then after 1 month, we observed the disease severity index. And we have seen that the Aureobasidium pullulans, either alone or in the pre-applications, they reduce the disease severity approximately 73% compared to the control treatment. However, we also did the gray mold against the Botrytis cinerea greenhouse experiment, but we did not find any significant effects to control this disease. That was quite interesting because, however, we tried to give answer why it is happening. So we come up that maybe the spray applications of spore suspensions, when we mix within the water, they increase the relative humidity, thereby optimizing the abiotic conditions for gray mold development. So we want to rule out this hypothesis. So we did the entomovectoring technology and collaborated with the bio-based company based in Belgium. And then we developed this powder formulation of the Aureobasidium pullulans and used the bumblebees to deliver this Aureobasidium pullulans onto the fruits of the strawberry crop. So we asked ourselves, does the application of pullulans using bumblebees lead to the biocontrol of the gray mold? If that's true, then how these bumblebees extend the shelf life of harvested fruit crop? So we designed the experiment based on the 6 comprehensive treatments. One was the controlled formulation, which is vectored by the bees, pullulans powder formulations vectored by bees, and pre-stop, which is commercially available product based on the Clonostochus rosea. And the 4th treatment was the control, no bees or no formulations. 5th was spray application based in the water. And the 6th was spray application of water only. So we divided this experiment into greenhouse just to avoid these cross-contaminations. So ripe fruits were harvested after 4 weeks, and we measured 6 gray mold disease. So we have seen that on the x-axis is a different combinations of the treatments, and the y-axis is a disease score scale 0 to the 4. And we see that A. pullulans, which is vectored by the bees, reduced the disease severity 69% when we harvested the fruits very freshly at day 0. While following 7 days of incubations, A. pullulans lowered the gray mold infection 73%, while pre-stop only inhibited 50%. A. pullulans 45% more efficient than at least during studied conditions. So we also measures the effect of the A. pullulans when vectored by the bees on the shelf life fruit. We have seen that when A. pullulans vectored by bees prolonged the shelf life 100%, and the A. pullulans vectored by bees showed first symptom after incubations at the day's 10th. However, A. pullulans either applied and vectored by bees or this by spray or pre-stop, they increase the shelf life compared to the control treatment. So we also tested that the A. pullulans under field conditions, and this was the trial based on the 2 years data. We harvested the fruits in different weeks, and we have seen that when A. pullulans applied in higher and the lower concentrations, they increased the shelf life approximately 36 to 24% compared to the control treatment. And we also included in this sugar beet extract, which is also effective, it's up approximately 16%. And we also tested the plant growth promotion effect that when we apply this Aureobasidium pullulans, at least at the later season of the strawberry crop, they increased the fruit productions 53% compared to the control treatment. So take-home messages. Application of A. pullulans enhanced the protection against crown and root rot gray mold. A. pullulans vectored by bees significantly reduced the severity of gray mold infected fruits and enhanced the shelf life when vectored by the bees 100% during post-harvest storage. Weekly application of A. pullulans and sugar beet extract reduced gray mold under field condition and increased the shelf life. And we also shown that application of A. pullulans indicates plant growth promotion effect on the plants. So with that, I would like to acknowledge, of course, collaborators and the funding agencies, and thank you for your attention. If you have questions, I would be happy to take that.
So thank you very much. We have some questions from the audience. That I've lost it. Okay. Okay. Does the timing of biological control application influence its success in preventing or reducing green mold outbreaks?
Yes, that could be possible that the timing of the application is very important when we're applying the biocontrol agents. But we did like 5 to 6 applications in each weekly basis, just to avoid this thing. So basically, I think this is, yes, timing is very important, of course, to find out. Yeah.
So another question: could bees transfer the product to another non-target plants?
This product transfer to the, what's the question?
The question is, could bees transfer the product to another non-target plants?
Yeah, that would be very interesting to test. Yes, we haven't done that actually. We only applied on the strawberry. We tested on the strawberry plants, but yeah.
Okay, thank you very much.
Yeah, thank you.
Shall we say, give people a chance to ask more questions? So there have been a lot of more questions have been asked in the chat. We'd highly encourage people to help each other answer these questions. But also, if the people have some burning questions to some of the other speakers, and I know that there were several questions in the chat, you're welcome to come up to the microphones and ask those questions. Now. Not in the coffee break. No, nobody brave enough, okay. But you're going to take the last slides. Okay. So we have a few slides to wrap up this session, and Sabrina will take those.
So, thank you very much. We are at the end of this first part of the biocontrol session. I would like to thank all the speakers that gave their contribution for this session and with their interesting talk. Thank you to the audience, because with this interactive modality, we have seen that there are several questions. Maybe the speakers can reply directly using the app during the coffee break, I don't know. And thanks to my co-chairs, Dan JENSEN and David COLLINGE. It was a huge work to select the oral presentation, but we hope to have done a good job. Just two reminds before closing the session. At 6 o'clock, we have 4 presentations, flash talk presentation during the poster session. But don't forget that we have almost 150 posters between the first and the second part of the biocontrol session. And again, don't forget tomorrow to attend the second part of the session from 10:30 in the morning. The session will be chaired by Monica Ofte and by Cléa Pligean-Combaré, and sorry for my bad French, this is my best. So thank you very much, and enjoy the coffee break.